restored and created from that system. Now, let me start out. We had a wonderful meeting last night where we were talking about the different functions of wetlands. Basically, why do we care about these silly things? Now, why do we go out and get our feet wet? Why do we get muddy? Why do we start mud all over our houses? Well, it's because of the functions these serve in the system. What I like to do, I like to talk about these functions, but I like to try to talk to them, talk about them as something that serves humanity. So, back in 1995, the National Academy of Sciences created a list of 9 functions that they felt were extremely important to humanity. Now, I broke these into 3 distinct parts. The first part is the hydrological part. And as you see here, first ecological function is short-term water storage. We're going to come back and talk about that in a second. Let's go on to the next. Long-term surface water storage, also a function. And third, maintenance of high water table. Now, I asked you as a group, you see me as a scientist, you see them as what? How do you take that and give it to a politician so that it makes sense? How can you express to them how important these things are? Think about that. We're going to come back and try to talk to it and figure it out. There's a second set in here, transformation psychology. There are 4 parts to this. I'm not going to have enough time today to talk about this. It's a whole different lecture, biogeochemical cycling. I will come back at the end and touch on it a little bit. But again, I really don't have time to go into that today. It's a very complicated process. The last 2 are 2 that I spend a lot of time with. And I'm going to use those to get to the first 3. And that is maintenance of characteristic plant communities. Again, I look at changes that take place in these systems due to stress. So these become, the last 2 become very important. And in fact, we use these to get to 1 through 7 as far as maturation of ecological functions. Okay, take a look at this table real quick. This is your short-term surface water storage, and again, this is kind of what I see when I look at it. Reduction of downstream peaks. Nice little diagram showing, of course, what happens with a wetland is you knock that peak down. We now know it's not just a function of delay of time, there's also evaporation, transpiration processes that take place, the delay of the water flow, etc. And what you're seeing is a decrease in that peak flow. Now, that's from a scientific point of view. What about the politician? Are they going to look at that and they're going to see that, oh, this has value? Again, we'll come back to that in a second. Let's look at the other 2. Again, just from a scientific point of view, short-term surface water, long-term surface water, and maintenance with high water. From a scientific point, again, this is what I'm going to see. What happens in those systems from an ecological perspective? Well, what if I did this? What if I turned around and started adding societal value? Now when I talk to a politician, I don't talk about short-term surface water storage. I talk about reduced property and crop damage. I talk about a lack of damage to life. I talk about its value to society. We can do that for the entire suite. We can talk about long-term storage as maintenance of fish habitat, biodiversity. We can talk about crops, increased production. Those are things that we can talk to the politician about. But let me ask you something else. How do we, as scientists, measure those things? That's a whole different question. So the concept is we can talk about functions, but let's move on and talk to the politicians in terms of society and humanity. Okay. One thing that came up yesterday, we're going to talk to you about. We have a lot of coastal wetlands in this area. Where does the water for Taipei come from? Your drinking water doesn't come from the incoming flooding tides. It comes from these seeps, and this is a beautiful little fen that I saw up in the mountain. These are the recharge sources for your groundwater. So it's as important, again, when we speak to the politicians, that they recognize it's not just local. It's going to be the adjacent properties as well, the watershed. You've got to get past those political boundaries. Okay? Now, I'm going to shift gears just a little bit. We're going to talk a little bit about habitat and food. Again, if we look at it from societal value, basically here, I look at this part of it here. I look at the changes. I see numbers. I see vegetation parameters. I see morphometrics. But if I'm going to translate that to a politician, I need to be talking about how it supports fisheries, aesthetics, how it does something for society, adds its economic value to that system. Now, how do we do that again? It's a complicated process. We are trying to do it through our assessment process. And I'm going to try to tie these together a little bit later in the talk. All right, so how did I get here? Well, like I said, I am a wetlands ecologist. I'm a botanist, basically, by training. I look at vegetation dynamics, the change in the assemblages over time, in response to stressors. Now, when I look at creating and restore wetlands, I'm looking at that same process. I'm looking at changes over time. The difference is, why do we restore and create wetlands? We're not just looking for changes. We are trying to restore the ecological functions that pay back to society. We are trying to create something for our society that is based in the ecological functions of these systems. So we have to go back and we have to measure, literally physically measure, parameters that we can use to explain maturation processes. In the United States, we call those ecological performance standards, and it's basically just looking at mathematical presentation of the biota in the system. And what I mean by that is very simple. You can go out, you can count parents, how many do you have in that area, or you may want to count the number of different species of birds, or you may want to try to count or capture a mud skipper. Actually, I fell in trying to get that one. But those little suckers run away from me. I can't catch them. I can't count them. So what do we do? Luckily for me, being a botanist, I've got the answer. Plants, they don't run away. They stay there. I can count them. I can pull a leaf off. I can taste them. I can do anything I want to them. They're not going to get me. So we look to these plants. In fact, in the United States and in most countries now, these EPSs are based on the morphometrics of plants. They're based on counting. They're based on looking at what is there today and monitor it over time and what changes take place over time. These morphometrics, however, have had some problems. Few of the ones that we've used in the past, stem density, the number of plants per unit area, the number of trees per hectare, for example. It's also a measure of survival. We also have stem and bowl area. Foresters talk about diameter at breast height, 4.5 feet or 1.2 meters. And we have stem area at ground level. Now, I'm going to come back to that SAG in a second. That turned out to be much more important than anybody thought. We also have things like canopy cover. Canopy cover is when you literally look at, go into a forest and look up and say, that tree covers 50% of this area. Now, I might go out there and I might see a tree covering 50%. Dr. Ott might go out there and he may see that tree covering 30%. Of course, he's going to be wrong, but how do we tell the difference? And what we've got to do is come up with something that is more accurate, so that Dr. Ott can go out, I can go out, we come up with the same answer. And that's where these morphometrics have failed. That's why we've had problems. Here we have measures like plant species richness, plant species diversity. Anybody know where diversity comes from? Basically, it comes from disorganization. So if you're measuring disorganization, extremely difficult to do, and we don't know what it means. We don't know how to compare diversity indices. We don't know how to statistically treat it because there's so much variance in diversity measures. So we've got some problems. We've got some problems that we know that some of these morphometrics we've been using for about 20 years literally have problems. There's too much variability. We're not getting the accuracy. We're not getting precision. So what are we going to do about it? we actually received a long-term grant to test a bunch of these EPSs. How are we going to test them? Well, we set up 7-hectare-sized plots. This is a very large experiment. We planted 7 different species of wetland trees. We treated them with different hydrologic processes. And then, 4 times a year, we went into this area and measured all of the morphometric parameters: stem density, stem height, tree height, number of tree branches, etc., etc. Now, this is a massive data set. We did this over 8 years. What it did give us was a massive data set that we were able to use to tease out the variables. So we collected the data from the control site, and then, using carbon, which is one of the most important ecological functions, carbon accumulation, we tested whether each of these morphometrics matched the carbon numbers that we had. We literally went out and took these trees. Some of these trees are 30 feet tall. Cut them down, dug up their roots. I have pictures of my students down 6 feet in the ground because they had to get all of the roots. Also, they take the tree and cut it up and weigh it, not drying it in a large greenhouse. So this was a very, very extensive process, but it gave us some great data. Again, it took us 8 years to get it all together. So we could compare that to, that gave us biomass, one of the most important aspects of function. And then our hypothesis was that by measuring the EPS, we were able to look at whether a restored or created system is replacing the loss of ecological functions. Make sense? So we went out, we measured a function, we went out and measured all the morphometrics, then tested the morphometrics against the known function. What we found, and I'll give you a little bit of orientation here, the p-value is a probability. We use a probability of 0.05, which means 95% of the time our data is not random. In this particular case, you see you have a p-value of 0.02, meaning there was a relationship between, and in this case, is biomass of carbon. I'm using it as a process of carbon here, and the percent survival, how many trees survive year after year after year after year. And what you see here, in the R square, is whether this variable actually explains the variation in this variable. The magic number here is we look for a minimum of 0.6. So we've got a mathematical number that we compare to, and what we look for is a p-value less than 0.05, which we have, but this does not meet the criteria for cause and relation, cause and effect. What that tells us is that this particular EPS, survival, does not predict the ecological function. Now, we went through all of them. I just picked a few out here. Here is percent cover. This is Marinus' number. All right, percent cover, and you can see what happens as they get older. It's not scattered. Okay, and again, we need it. There is a relationship, which is interesting. We're going to come back to that in a second. But at the same time, it doesn't meet that 0.6. So it's not a cause and effect relationship. So something's driving them together. They're growing together. But cover is not going to explain your ecological function. Last, or as Kim would say, lastly, we look at canopy cover. And as you see, the smaller trees, these are earlier growing trees, are not, they're pretty tight. Here I've got again, the p-value is significant, but I still don't make that 0.6. It's close. It's close. But look what happens. As the trees get bigger, they get farther and farther apart. And as we went back to the 9th year and actually measured them, that kind of fell apart. It's actually not part of the study, so I didn't put it here. Okay? So that doesn't work either. Again, we're seeing the relationship, but we're not seeing the cause and effect. Okay, so what does that mean? Well, the one that did fit into this was this SAG, S-A-G, stem area ground line. Here,